Today we will learn about food-borne animal parasites. According to World Health Organization, food-borne diseases are defined as any disease of an infectious nature or toxic nature which is caused by the consumption of contaminated food or water. Major causes of food-borne diseases include microbial agents like bacteria, fungi, algae, virus, etc. and they occur primarily due to lack of sanitation or proper hygienic conditions. And these diseases affect mainly the developing countries. Some of the examples of food-borne parasites are parasites which include on other organisms which is known as the host for food, and they utilize the host tissue for their own reproduction, and after their reproduction and multiplication they are excreted along with the feces of the host organism to complete their life cycle. Around 300 species of parasitic worms are reported to be present in human gut. Food-borne parasitic diseases are of major concern for human health. Most common examples are Giardia, Cryptosporidium, Cyclospora, fluke worms, tapeworms, etc. The food-borne diseases incidences are increasing due to globalization of food supply and increased international travel, which is exposing people living over the different continents. Common food-borne animal parasites are protozoa, nematodes and helminths. These parasites may be either single-celled or multicellular, and their mode of transmission is from animal to human, from human to human and from human to animal. Some examples of common food-borne parasites are beef, Taenia saginata, which is a tapeworm. Pork and other meat will have Toxoplasma trichinella. Milk will have Cryptosporidium. Fish and marine animals will have Gnathostoma, and fruits, vegetables will have parasites like Cyclospora, Cryptosporidium, Giardia, Taenia solium, Fasciola, etc. And water also harbors some of the parasites. These are Cyclospora, Cryptosporidium, Giardia, Toxoplasma, Ascaris, Fasciola, etc. The life cycle of the parasite includes a host which allows them to multiply. Now, let us know some of the diseases caused by protozoa. The prominent examples are amoebiasis, giardiasis, toxoplasmosis, cryptosporidiosis and cyclosporiasis. The symptoms of this infection are diarrhea, abdominal pain, vomiting, etc. Bloody diarrhea accompanied by mucus due to ulceration of the colon is a common symptom. And along with feces, the cysts of the protozoa are shed. Sometimes there are complications due to invasion of other tissues like liver, and it may also cause appendicitis and abscesses of liver, lung or brain. The disease can be diagnosed under laboratory conditions by detection of trophozoite and cyst in the stool sample. Microscopic examination is a common method. Sometimes specific antibodies in the blood are also used to detect the amoeba, DNA and protein in the feces. The disease can be prevented by maintaining proper personal hygiene, avoiding contaminated water and food which is contaminated with human feces, and chlorination to kill the viable cyst in the water. There is other disease caused by protozoa which is known as giardiasis. It is a diarrhea disease caused by Giardia intestinalis or also known as Giardia lamblia. It also survives as cyst in the food and water, and it is a common epidemic outbreak in many countries. It primarily affects the children, especially children in daycare centers. The major source of transmission of cyst is contaminated water supply. The symptoms appear after an incubation period of 1 to 2 weeks, and these include acute or chronic illness, diarrhea, epigastric pain, cramps, anorexia, bloating, flatulence, etc. The disease can be diagnosed in the laboratory conditions by identification of cyst in the stool samples and identification of trophozoites in case of severe diarrhea. ELISA kit are also available for detection of the pathogen. There is no need for any specific treatment because the infection subsides on its own. In case of severe infection and if the symptoms persist, the drugs may be given. The disease can be prevented by adopting adequate sanitary measures like proper treatment of community water and destruction of the cyst using standard cooking procedures. Toxoplasmosis is another protozoa mediated disease. It is caused by Toxoplasma gondii and it is very much prevalent in developed countries as well as in other countries. In majority of the cases, it is the infected animals, birds, domestic wild cats which are the host for this protozoa. Especially the cats are the primary host because the protozoa requires completion of the sexual cycle in this host. The transmission to humans is primarily by ingestion of oocyst directly from soil or water which are contaminated with cat feces, or through consumption of raw or undercooked meat, especially pork and other such products. The symptom of the diseases, there are no special symptoms and mild flu-like illness in healthy individual is generally seen. However, in immunocompromised individual, infection is life-threatening. The protozoa can be diagnosed under laboratory conditions by serological test to detect humoral antibodies include ELISA, and detection of antibodies against Toxoplasma gondii is in one serum sample only ascertains that the individual has been infected at some point of time in the past. It is advised to collect 2 samples from the same patient after an interval of 2 to 4 weeks. The disease can be treated by a use of a combination of 2 drugs, sulfadiazine and pyrimethamine. The disease can be prevented by using properly cooked meat and raw eggs, washing the hands properly after working in soil, and keeping the household cats indoors as much as possible. And it is also advised to clean the cat litter box daily and to feed the cat with commercially available diet. There is another disease caused by protozoa that is known as cryptosporidiosis. The organism which is responsible for this disease is the protozoa Cryptosporidium. Cryptosporidium parvum is the first causative protozoa identified, and it is the most commonly detected in majority of the human cases. The symptoms appear after a period of 5 to 28 days, and the common symptom is diarrhea, which is similar to that found in cholera. It is also accompanied by abdominal pain, nausea, vomiting, fever and fatigue. The protozoa is identified or diagnosed in the laboratory by microscopic method, staining method, and by using antibodies specific for it. Routine diagnosis by concentrating the fecal matter or oocyst in the stool is commonly used method. Advanced techniques such as fluorescent antibody techniques can also be used to detect the presence of specific antigen oocyst of Cryptosporidiums, and it is highly resistant to chlorine and many other disinfectant. Therefore, their inactivation during the water purification process is a major challenge. There is no specific therapy available, and some drugs may suppress the infection, and the most common and important factor is rehydration with electrolyte replacement therapy. The disease can be prevented by chlorine dioxide and ozone treatment of water, which may inactivate the parasite. Low doses of ultraviolet light exposure can also inactivate the oocyst. The safest method to decontaminate the drinking water is to boil it. Cyclosporiasis is also a disease caused by protozoa, and it is endemic in tropical and subtropical environment, although its presence has also been reported from most of the countries. The outbreak occurs by consumption of contaminated soft fruits and vegetables. The largest foodborne outbreak to date has been associated with this parasite, Cyclospora cayetanensis. The common symptom of the protozoan infection is ingestion of oocyst, and the symptoms range for a period of 2 to 11 days. The common symptoms are non-bloody diarrhea, anorexia, emaciation, nausea, vomiting, fever, fatigue, cramps and bloating. The disease can be diagnosed in the laboratory like all other protozoan based on the identification of oocyst in the feces. It is advisable to observe several stool samples over a period of time for accurate diagnosis. The disease can be prevented by administration of a combination of drugs trimethoprim and sulfamethoxazole. The fluid must be involved in medication to compensate for the water loss during diarrhea. However, at present there is no vaccine available to control the human infection. To prevent the infection, the food should be cooked thoroughly and the water should be devoid of the contamination with Cyclospora oocyst. There are also certain diseases caused by helminths. These are transmitted to human by food and water. Although the helminths do not multiply in the food, but they can be detected by microscopic examination. Some common helminths are fascioliasis, which is caused by Fasciola hepatica, which is also commonly known as the liver fluke. It infects a variety of mammals and it is found in different parts of the world. The parasite can reach a length of 30 mm and a width of 13 mm, and this is one of the largest fluke reported. The incubation period after the liver fluke infection is of about 30 days, and the symptoms appear as fever, malaise, fatigue, loss of appetite and weight. They also may cause discomfort in the liver. The diagnosis under the laboratory condition can be done by microscopic examination and detection of eggs in the stool of the infected individual. The drug which is recommended for the treatment is Rifaxidenide, and it is commonly used for treatment of the disease. The prevention of the disease can be done by avoiding contact with contaminated foods and to maintain proper hygiene. There are other diseases caused by helminths. These are teniasis. Teniasis is caused by Taenia saginata and Taenia solium, which are the common flatworms causing teniasis. The first one is known as the beef tapeworm and the second one is known as the pork tapeworm. These infect pigs and human. Taenia solium infection is highly dangerous than Taenia saginata. The parasite is normally 2.3 to 3 meter long. Larval stage causes cysticercosis. The disease is highly prevalent in underdeveloped countries where poor sanitation practices are prevalent. The symptoms, diagnosis and treatment is the incubation period for the beef tapeworm is around 10 to 14 weeks, whereas it is 8 to 12 weeks for the pork tapeworm infections. The teniasis is usually asymptomatic, but the symptoms may differ in human host. The infected person may experience abdominal cramps, weight loss, digestive disturbances, gastrointestinal obstructions. They also cause irritation in perianal area. Laboratory diagnosis of this parasite is by means of the presence of eggs in the stool of an infected individual. Albendazole is the common drug of choice for treating the disease, and the disease can be prevented by avoiding consumption of undercooked pork and by maintaining proper hygiene. There are also certain diseases caused by nematodes. These nematodes are generally transmitted to human being by food and water. They do not multiply in the food and can be detected by microscopic examination. Trichinosis is caused by a species Trichinella spiralis and it is present in undercooked pork. The incubation period varies from 1 to 2 days to as long as several weeks, and it produces symptoms like nausea, vomiting, diarrhea, sweating, abdominal discomfort and loss of appetite. When the parasite moves to the skeleton muscles, the person feels muscular soreness and swelling. In severe cases, the death may also occur. The detection of ovoid capsular cyst in skeletal muscles and antibodies in serum of an infected individual is a common laboratory diagnostic test. The disease can be prevented by avoiding consumption of undercooked or raw pork. The destruction of the parasite could be achieved by cooking the pork at high temperature, that is at least 58.3 degrees Celsius, quick freezing or storage at minus 15 degrees Celsius. The treatment involves the use of drug mebendazole or albendazole. So, let us summarize that foodborne parasites are one of the major causes of foodborne diseases. The important feature is that these parasites do not multiply in the food as such, they multiply in a live host. There are different groups of parasite right from protozoa, helminths, etc. These are causative agent of numerous diseases, and some of these diseases can be life threatening also. In order to avoid the diseases, it is essential to follow sanitary practices, maintain good hygienic conditions and properly cooking the food in order to avoid these pathogens. Thank you.